Hi everyone, welcome. So in this video what we're going to do is we're going to really tie everything that we've learned about the immune system together, and we're going to look at how we have developed medical interventions and treatments by manipulating and using the immune system to try and treat diseases. So when we make medical interventions that involve the immune system or manipulation of the immune system, we call these immunotherapies. So immune and then therapies, immunotherapies. So a large part of the prevention of disease is in the form of these sorts of treatments. So this is any therapy that changes the way the immune system functions. And there's two different types of immunotherapies that we're going to go through today. So we can have activation immunotherapies. So this is therapy that aims to activate an immune response or induce or amplify an immune response. And then we have suppression immunotherapies. This is where we have therapies that are trying to suppress or reduce or prevent an immune response. So depending on what kind of disease it is, you might want to amplify an immune response or you might want to suppress it depending on what it is. The little table here, you don't need to know, but it's good to know a couple of different of the therapies that are currently being worked on right now that are looking really promising and really cool. We're going to learn about one today, which is not in this table, but these are a couple of other examples of treatments that are, currently using the immune system to treat diseases right now. So the one we're going to learn about today is something called monoclonal antibodies. Okay, so what are monoclonal antibodies? So mono means one. Okay, so one type of antibody to one specific thing. Clonal means clones and antibody means antibodies. So what these are is it's antibodies that are produced in a lab that bind to a specific antigen. So we'll take one specific antigen we're interested in and then we'll make many clones of plasma cells to produce many clones of antibodies that are going to detect and attach and destroy or, suppress a specific antigen or target. So you might see them referred to as MABs, and I might refer to them as MABs in this video. So we're going to learn about what monoclonal antibodies are, how they're made, and what they can do. But basically, they can do pretty much everything normal antibodies do, which we learned about a few videos back. It's just that the way that we make them is a little bit different so that we can streamline it and make them in a lab and make them a bit more targeted. So this is a big slide, but this is how we produce monoclonal antibodies. And I've got it in a step-by-step form, and I've also got it in a picture form as well, so whichever one works for you. So the first thing that we're going to do is that we are going to identify and isolate an antigen of interest. Obviously, we need to know what we are targeting, so we need to identify that antigen. And that antigen could be anything. It could be a specific antigen on a cancer cell we're trying to treat. It could be an antigen that's on a specific cell type. It could be a range of different things, but we need to identify and isolate that. Then what we're going to do is we're going to vaccinate an animal. It's normally a mouse or a rat. We're going to vaccinate an animal with that antigen. So, and the exact same thing that happens in vaccinations in us is going to happen. So when we vaccinate that mouse with that antigen, it's going to stimulate an immune response. So it's going to stimulate the selection and proliferation of B cells, and it's going to be specific for the antigen that we have injected into that mouse. Then what we're going to do is we're going to extract the B cells from the mouse. Why B cells? Because we're trying to produce antibodies, and we know that it's the humoral B cell immunity that creates those antibodies, so we're going to be quite specific in targeting B cells. So what we're going to do is we're going to extract those B cells that have been selected and expanded and differentiated, and we're going to take them from the spleen of the animal. Why the spleen? Because that's a secondary lymphoid organ and that's where our lymphocytes normally reside. So we're going to take them from the spleen. Then what we're going to do is we're going to take those B cells and we are going to fuse them with something called a myeloma cell. So a myeloma cell is actually a cancer cell, which sounds a bit, counterintuitive, but what they are is they are rapidly dividing cancerous cells. And obviously, they're not going to hurt anything. We strictly use them so that our B cells can divide more rapidly. So when we fuse these two things together, they create something called a hybridoma. So this is a B cell and a myeloma cell together is called a hybridoma. And this means that the B cells can grow more quickly and they can divide more quickly and we can make really large quantities. So that's what we do next. And then what we do is we take those hybridomas and we screen them so that only the cells that are have the appropriate antibody are selected, because we know that our plasma B cells can make a different range of antibodies. We've got IgE, IgM, IgG. So we want to select the ones that are really specific to what we want them to do. And then what we're going to do is we're going to clone and mass produce that antibody. So we want massive amounts of a specific type of antibody. So there's going to be many clones of that antibody, and then we're going to collect them, and they're going to be used to treat the patient for whatever they have. So this is also demonstrated in this diagram here. So we isolate the antigen, we vaccinate a mouse, we extract their B cells, we fuse it with a myeloma cell, and then we mass produce the antibodies that we are after. So what can we do with these antibodies? There's a couple of different things that we can do with these antibodies, and I'm going to give you two different examples of what they are. So one thing that we can do is we can use them against cancer. And you're probably all very familiar with cancer. I certainly am. You probably know someone that has suffered. So this is obviously a major disease that we're always working on to try and improve treatments for. So it's a complex group of diseases, but it's caused by uncontrolled and unregulated replication of mutated cells. So cancer has been around for a long time, and as a result, it's evolved, and it's been able to evolve to evade the immune system. So what that means is it can display or not display certain molecules. So they've actually gotten really smart so that they can evade the immune system and they won't be detected by T cells or B cells, which is really quite tricky. So what we've done is we've developed some immunotherapies that can tackle this, and monoclonal antibodies is one of those immunotherapies. And this would be an activation immunotherapy, because what it's trying to do is it's trying to help the immune system to recognize and destroy cancer cells. So we're trying to activate the immune system, and monoclonal antibodies can do this. So a couple of the different things that they can do, and hopefully these are looking quite familiar to you because they're all really similar to the kinds of things that antibodies naturally do in the body anyway. The only difference is that we've pinpointed the exact target that we want it to destroy or mark. So it can do antibody-dependent cell-mediated cytotoxicity, which is a long word. We can shorten it to ADCC, but basically what this means is that monoclonal antibodies can bind to cancer cells, which can attract natural killer cells to kill it, because we know natural killer cells are really good at targeting infected cells, so they can bind and act as little markers for NK cells to come over and kill it. We can also activate complement, so we know that antibodies can bind to pathogens, and this can help to promote the formation of a membrane attack complex, and this can help to induce apoptosis or lysis in cells, which can kill cancer cells and also attract immune cells to wherever they need to be. There's also a couple that are specifically unique to monoclonal antibodies. So they can also do something called checkpoint inhibition. So this is so there's a certain amount of immune checkpoints that are natural regulators of immune function. So all cells will go through these various checkpoints to make sure that they're, not killing healthy cells or anything like that. What cancer cells do, because they're quite smart, is also secrete molecules that stimulate these checkpoints, so the immune system has to stop, and they don't actually have the ability to recognize cancer cells. So what monoclonal antibodies can do is they can bind to these checkpoints that can slow down immune responses, and therefore the cancer cells can no longer avoid, detection from the immune system. And one that's also specific to monoclonal antibodies is that we can actually attach drugs to antibodies, and these are called conjugated monoclonal antibodies. So we can attach things like chemotherapies, for instance, and if we attach them onto antibodies, that means that chemotherapies are only going to the target areas, and we can prevent all those really nasty side effects that chemotherapy normally has, because it's normally quite widespread and doesn't have a target. So we can specifically target cancer cells with those sorts of drugs, which can help with symptoms and can also kill cancer cells more effectively. Monoclonal antibodies can also block cell growth and trigger apoptosis as well. So these are the sorts of things that monoclonal antibodies can do to activate immunity, specifically against cancer. But we know that monoclonal antibodies can also suppress immunity. So we've got the two different types. And an example of suppressing immunity and why that might be good, because normally we would think we don't want to do that because we want the immune system to do well. Well, an example of when we'd want to do that is in autoimmune diseases. So remember, we learned about autoimmune diseases a little bit in our first video. What these are is when the immune system recognizes self cells as non-self cells, and they therefore attack self cells. So obviously this is a problem where we actually want to suppress the immune system, and normally B and T cells become really, really reactive, and they'll produce autoantibodies, and T cells will start to attack our self tissues, and this is very, very bad. So we can actually suppress the immune system, which would therefore help the treatment of these autoimmune diseases. So we can do things like monoclonal antibodies can inhibit cytokines. So we can make a monoclonal antibody that's really specific to cytokines, and they can bind to the cytokines and stop them from doing their jobs and activating immune cells and things like that. We can also create monoclonal antibodies that can bind to B and T cells, and this binding to specific molecules can actually stop them from functioning or inhibit them, or even mark them for destruction by other immune cells. And we can specifically target the autoreactive B and T cells, so that only the ones that are causing the damage are going to be damaged, because obviously we still need B and T cells for other diseases. So we can be really specific in that way. So monoclonal antibodies is a really good example of how we can use activating and suppressing immunotherapies to help target diseases and manipulate our immune system to help to stop various diseases. So hopefully that helps. Please bring any questions to class, and I will see you there.